Hello, we are Amina and Misa from the Period 4 AP Bio class, and we will be talking about the adrenaline pathway that affects heart rate or lung capacity. Specifically, we will be explaining how the signaling and receptor proteins work, as well as the specific workings of the pathway and termination from the players to the process from start to finish.
As a hydrophilic peptide hormone, adrenaline can't move across the hydrophobic interior of the cell membrane. Instead, it binds to cell surface receptors. Beta-adrenergic receptors are proteins found on the surface of cells that respond to the hormone adrenaline that are a type of GPCR protein. GPCR proteins are the largest family of membrane proteins and mediate most cellular responses to hormones and neurotransmitters. They're transmembrane proteins that play a fundamental role in signaling. Its purpose is to transduce extracellular information into intracellular functional responses. When a ligand binds to a GPCR, it can be used to trigger a complex chain of events involving G proteins and second messengers in order to bring the message to the inside of the cell and influence various cell functions, which will be further explained later in the video. An advantage of the GPCR is that it acts as an entryway for signals and hormones to relay information into the rest of the cell. It also creates quick response. However, this means that it is quick to shut down, which means that the response does not last for a long time, which is a disadvantage.
Adrenaline, or epinephrine, binds to the beta-adrenergic receptor, which leads to the transformational change in the GPCR domain, with which the GDP, bound with the G-alpha protein, is replaced with GTP. This process is mediated by GEF, which is Garnasi nucleotide exchange factor. The binding of GTP with the G-alpha subunits drives the activation of the G protein. G-alpha then dissociates from the rest of the G protein, and it binds to adenylyl cyclase. This causes the adenylyl cyclase to become activated and go through conformational change in shape, which then mediates the conversion of GTP to cyclic AMP, which is a second messenger. Now that the original signal has been relayed into an intracellular signal with CAMP as a second messenger, the original message is connected to the response through a series of proteins. The CAMP molecule attaches with the inactive form of protein kinase A and activates it. Inactive phosphorylase kinase is made into active phosphorylase kinase. Then this phosphorylase kinase acts upon the inactive glycogen phosphorylase enzyme to make it active. The active glycogen phosphorylase acts upon glycogen and mediates the breakdown of glycogen into glucose 1-phosphate, which is used in metabolism later in different pathways. A muscle cell therefore rapidly gets a large, ready pool of glucose molecules, which then becomes available for use by the muscle cell in response to a sudden surge of adrenaline, the fight or flight metabolic response.
In order for a cell to respond only when a signal is present, the players in the pathway have to be regulated so that they are activated for only a short period of time. In order to do this, the termination process is needed. When the external signal is no longer present, the intracellular signal is terminated. First, the alpha subunit from GTP to GGP. After it makes CAMP, the alpha subunit from the G protein comes off the adenylyl cyclase, causing it to go back to its inactive form. The inactive GDP bound to the alpha subunit dissociates from the adenylyl cyclase, attaching back to the rest of the G protein. This prevents further activity of adenylyl cyclase. Then, an enzyme called phosphodiesterase breaks the CAMP into AMP, which decreases the concentration of CAMP, and protein kinase A becomes inactive. Various protein phosphatases cause dephosphorylation of the protein that was phosphorylated initially. This reverses the activity of that protein. All of these contribute to the termination of the signal and response.